Okay, so let me begin. Let's see if I can. Do you guys? Am I going forward? Okay.
Yes.
Okay. Thanks. So I think as anybody who studies biological systems has come to appreciate, biological systems. It doesn't matter if one's talking about proteins, cells, or ecosystems. They're generally considered very complex, and a big reason people use this term is because the way that we parameterize or describe these systems is in a space that's highly entropic, whether that is the set of amino acids that correspond to what defines a protein, the set of genes that corresponds to the genome that defines a cell, or the set of taxa that define the microbial ecosystem that one is studying. But what's also worth noting is that though these systems are remarkably complex, they're also really well ordered. So that is to say that proteins adopt a very specific fold and execute precise functions. Cells have compartmentalized organelles that are able to perform very specific functions. Ecosystems, though we don't have the same sort of descriptors like fold and function or compartmentalization at the scale of ecosystems, you can visually see how there's collective behavior that emerges from this high entropy of parts. And another thing that's worth noting is that there's no brain of these systems. They ascribe by what's called decentralized regulation. That is that the order that you're seeing on the screen arises from emergent phenomenon. Parts interact together to create subnetworks, and those networks interact together to ultimately create the kinds of collectives that we see. So from lots of parts emerge structure and associated function encoded within that structure. And a question that our lab is really interested in is how do we understand how to describe emergent relationships of systems? Now, for physics-based designed systems, this is actually a well-posed problem that has a really good solution, and the solution is pretty simple. You elucidate all of the parts and you look at the pattern of interactions between the parts. So here's a simplistic circuit, and we can write down explicit equations that describe the predictability of the behavior of this circuit based on the fact that there are 3 resistors, 2 of which are in parallel, 1 of which is in series, and therefore you are able to understand how this system works. The nice thing about these kind of theories is that they scale very well. So even if you increase the number of parts from, say, 4 to on the order of hundreds of thousands of parts, like in a Boeing jet, you can still, with minute-to-minute precision, be able to say, well, the plane's going to take off at one place and land in another place. And the entire reason for this is because there's a fundamental theory of a wiring diagram that we understand from engineering principles. Now, how do we understand systems in biology? So here's an example of one strategy for doing this. We take a genome and we perform genome-wide knockout screens. So we knock out one gene at a time and try to understand what the knockout does to the phenotype of, say, an organism. And with the advent of sequencing and the ease and accessibility with which sequencing is now being more cost-effective, we're really good at being able to identify parts of complex biological systems. But what about the pattern of interactions between the parts? And I say that I would argue that we're not very good at this yet. And the problem resides in the complexity of the possible interactions in biological systems. So if we take into account, for instance, all possible interactions within a very simplistic pathway made of 4 proteins, given 4 parts, the complexity, the possible number of interactions between these proteins scales as 2 to the 4. So here are all of the possible interactions that one has to now assay in order to see how individual proteins collaborate together to create complexes or pathways. Now, why do we have to do this? Well, it's because we don't have a good sense of what the wiring diagram of biological systems are, because they've come through the process of evolution. Iterative selection and variation is a totally distinct design process than, say, being able to put together parts in a circuit. Currently, there's an approach which is to intuit higher order interactions from lower order interactions. So in other words, patch together pairs of proteins that interact to create protein complexes or intuit pathways. But more and more as genome-wide studies become more high throughput and more deep, what we are finding is that there's de novo new information in higher order interactions between proteins. So there was a great example of this that was published in Science a few years ago, where in yeast, this group took a unbiased approach of being able to probe three-way interactions genome-wide in Saccharomyces cerevisiae, and what they showed was that you couldn't predict three-way interactions from all combinations of two-way interactions, meaning that there was intrinsic information in the three-way interaction terms. So combining all of these observations together, I think we arrive at a fundamental problem, which is that we need a model for emergence. That is, we have to be able to scale from pairs of interactions to higher order interactions. And we need to be able to do this in some sort of general way that doesn't require infinite resources. Imagine one now has 100 proteins in the pathway. This becomes a completely intractable problem experimentally. So this problem forms the central question of our work. How can we create a model of emergence given the constraints that we have with experiments? And one of the central hypotheses that we had was that perhaps evolution can help us with respect to understanding what interactions are salient to encode function at all scales of organization in biology. So here I'm showing you a picture of an organism, E. coli, that is comprised of cellular pathways. Each cellular pathway is comprised of protein complexes, and each protein complex is comprised of proteins, which are comprised of amino acids. And so the posit is that evolution has selected for interactions within parts comprising these different scales at each scale of organization. So what we thought was that maybe the pattern of genomic variation across biodiversity can tell us about not just any particular scale of interactions, but a hierarchy of interactions. That is how you get from, say, individual proteins to pathways. So what I'm going to tell you about is our strategy to try to answer this question. And what we decided to do was focus on bacteria as a model. And the reason for that is because bacteria have been sequenced immensely. So there's a lot of existing sequences that exist, the existing sequences from which we can pull. And the second is that we have ground truth better than most other organisms like viruses or eukaryotes with respect to interactions between proteins and whether those interactions are physical by nature, indirect by nature, or form a pathway. So using bacteria as a model, what we did to try to address this question was to download the non-redundant UniProt database of bacterial strains. In total, this was 7,047 proteomes, bacterial proteomes. And we annotated each of these proteomes by what are called orthologous gene groups, OGGs. An orthologous gene group one can think of simply as a metric for how functionally similar to units, genomic units are in a proteome. Now, importantly, there's no function that's loaded into defining OGGs. It's only based off of hidden Markov models of amino acid sequence identity. So if two proteins have very similar sequence identity, one could imagine they get collapsed into the same OGG. And the reason that we decided to do this was because this enabled now to make an alignment across the 7,000 organisms. Now, the order of the columns here doesn't actually make a difference, right? But what this enabled was a direct comparison of proteome I with proteome J. So the way to read this matrix is every pixel in this matrix is the number of orthologous gene groups in a particular species. So for instance, there are two COG 1283s in Celomonas, in this Celomonas species. And as a sort of goal, what we wanted to know was, does the statistical variance within this matrix map in any way to biological information characterized across the hierarchy of scales? And so the way we defined a hierarchy of scales was phylogenetic information spanning phylum down to genus. So whether two bacteria share phylum, class, order, family, genus, or indirect interactions amongst proteins within a proteome, or direct interactions within two proteins within a proteome. So one can think of this as sort of an exploratory target at the moment. So the first thing that we did was to probe the structure of this matrix. We performed a technique called singular value decomposition. For those of you who aren't familiar, it's a generalization of principal components analysis. Same idea, which is that you deconstruct a centered and standardized matrix in order to get an eigenspectrum, a spectrum of principal components. That spectrum is ordered according to statistical scale. So the first principal component, or SVD component, holds the most amount of data variance, and the last one holds the least amount of data variance. Now, this is what we found. This was the SVD spectrum of the orthologous gene group matrix. And what we see is something somewhat interesting, that the top component holds on the order of 6% of variance. And we see a linear scaling from the top component down to approximately component 3000. Now, typically, what we do when we see such spectra is we take the top components of variation and we discard the rest, and we have some model about what is a noise cutoff and what is not. And I think most of us would probably say that if we look at something like principal component 3000 here, which is on the order of 0.001% variance, that's noise. So what we wanted to do was probe this, interrogate this spectrum for the biological information shown here. So the first thing that we saw was that the top components of variation hold signal regarding whether or not bacteria share a phylum. Now, I don't think that this is completely surprising. I think this is well known that the broad components of data variance will share some very broad characterizations of bacteria whenever one does such a phylogenomic analysis. And we confirm this. If we look at SVD 1, 2, 3, and 4, you can see that the bacteria, which are shown in dots here and colored by phylum, basically cluster by phylum. But what was interesting was that the top 4 components only held 17% of variance. So there's a substantial amount of variance left in the spectrum that remains unexplained by just looking at the top components. And so what we wanted to do was then ask, well, what about deeper components? Is there any biological information contained beyond component 4? So I'm summarizing a lot of data here, but this is what we found. So as we went down the spectrum, we asked how much mutual information is there between the position in the spectrum and information contained in these benchmarks. So what are these benchmarks? NCBI phylogenetic classification from phylum down to genus, the string database, the Go database, gene ontology, and then 3 databases of whether or not 2 proteins interact with each other directly. CoEv Plus, which is an amino acid coevolution metric published by David Baker's group in 2019, the PDB, which is a metric of crystal structures where you can see co-crystal structures and actually see physical contacts, and EcoPsych, which is a database of affinity purification mass spec data. So the way to read this graph is on the y-axis, the mutual information, a measure of mutual information, and on the x-axis is spectral position, meaning where we are in the eigenspectrum. And the color key here matches the color key here. Now, when a curve gets to a value of 1, what that means is that deeper components no longer hold any more information about that benchmark. So what we found, if we look at a holistic picture of all of the benchmarks across the eigenspectrum, we found a really interesting trend. That is that there seems to be an order to the way that this information is stored within the eigenspectrum. First, we see information relating to phylum, then class, order, family, genus. Then below genus, what we see are information related to indirect interactions contained within the string database or the Go database. And then below that, what we see are direct protein-protein interactions, meaning physical interactions, contained within the databases of CoEV, PLUS, PDB, and EcoPsych. I think something that's worth pointing out is that irrespective of the type of database we use, for instance, if we compare PDB with EcoPsych, PDB is co-crystal structures, EcoPsych is affinity purification mass spec, we see an overlap in the information. And CoEV Plus is purely computational, right? So we see an overlap in the information suggesting that it doesn't really matter what database one uses, as long as there's robust enough information in that database, we're seeing the same sort of spectral filtering with respect to that information. So this led to the first result that we found, which is that the SVD spectrum of OGG variation across these 7,000 bacteria reflects a hierarchy of biology. That is, the top modes are enriched for broad phylogenetic information, and the bottom modes are enriched for protein-protein interactions within a proteome of an organism. So one of the typical problems with respect to protein-protein interaction inference is that when you try to infer based off of covariation or co-occurrence or any type of co-proximity, then it's difficult to disentangle whether or not a protein-protein interaction is phylogenetic, whether it's functional and not physical, or whether it's physical. And so given that the spectrum actually separates these different scales of interaction classes, we thought, well, could we predict indirect and direct interactions just from the statistics of coevolution? So the way we did this was by leveraging the fact that the biological information that we tried to find across the SVD spectrum is spread heterogeneously. So for instance, what we found is that phylogeny is mostly enriched in components 1 through 33, that indirect protein-protein interactions found in string and Go are mostly enriched from components 34 to 223, and for direct from 224 to 3000. So we defined a metric for protein-protein interactions called spectral correlations. And this is, one can think of this as a statistical proxy for whether or not two proteins statistically interact with each other. Now, what do we mean by spectral correlations? Here I'm showing you an example of two proteins, ARG-A and ARG-H. Now, you should know from the names that both of these are functionally implicated with each other. They're both involved with arginine biosynthesis, and they are comprised of the following OGG structures: COG 0548 and 1246 for ARG-A, and COG 0165 for ARG-H. So the way that we computed protein-protein, quote, spectral correlations is the following. Each OGG projects along the components of the SVD spectrum to a certain degree. So what we're showing here is the profile of the projections of each of these COGs onto components 1 to 200. And you can view this as sort of like a projection profile of a particular COG. What we then did was simply average the, if a protein had multiple COGs, we averaged the contribution across the components for the set of COGs that were within a protein. And so now we get an approximated protein projection on components 1 to 200. And then to compute the protein-protein spectral correlation between the two, we simply compute the Pearson correlation across this span of SVD components. So for instance, here, what I'm showing you is an overlaid profile of the approximated protein projection for ARG-A and that of ARG-H. And if you compute the Pearson correlation coefficient between the two, you see that it's greater than, it's positive, indicating that these two non-randomly statistically interact with each other from component 1 to 200. So that is what we mean by spectral correlations between two proteins. And the strategy here was to use protein-protein spectral correlations as features to input into a random forest model to be able to predict interaction classes. So we focused on E. coli K12 because it's the most studied bacterial organism where we have good ground truth. We created a gold standard of E. coli K12 PPIs where there were 72,000 non-interacting protein-protein pairs, approximately 1,200 indirectly interacting protein-protein pairs, and 72 direct physical interactions between proteins. We split this gold standard up into a 60% and 40% training and validation data set. And the workflow looked like this. So for protein pairs, we computed spectral correlations across these different windows where 1 to 33 was enriched for phylogeny, 34 to 223 for indirect interactions, and 224 to 3000 for direct interactions. So to give some intuitive sense of what this means, for instance, here, we know that RPMC and RPMIV are parts of the 50S ribosome. They physically interact, or they indirectly interact with each other. And what you can see here is that there's a high correlation, spectral correlation from 1 to 33 and 34 to 223, but lacking in high correlation from 224 to 3000. Whereas two parts of physically interacting proteins of nitrate reductase, you can see that there is a uniformly high spectral correlation across all of these different windows. And for those that don't interact, you see basically that the spectral correlation across the three windows is negligible. So then what we did was we know the true type of interaction. And so we can train an RF model based on this to be able to predict what the interaction is, and then test the fidelity of these RF models on the validation data set, which is an orthogonal data set to the 60% training set. We then validate based on the true type. So we compare the prediction and the true type by what's a metric called an F-score, the harmonic mean of precision and recall. So I'm going to summarize the comparison that we've done with our method versus a litany of published methods involving experiments and computational inference, and a PCA-based method that basically just takes into account the top modes of variation every time and extends the consideration through deeper and deeper into the spectrum. So for instance, modes 1 through 5, 1 to 10, 1 to 20, all the way down to 1 to 7047. So what we find is that for non-interacting data sets, all of them, non-interacting classes, all of them do quite well. And this isn't completely surprising given that there's so many non-interacting proteins in the E. coli K12 proteome. What is interesting is if we start looking at the F-score distributions for predicting indirect protein-protein interactions and direct protein-protein interactions. So the first thing to note is that compared to all the other methods, our method does the best across both classes. And to give a sense of what this means sort of intuitively, a great comparator is by canonical PCA approaches. So if we look at what happens as a function of adding in more modes of variation, we see a trade-off between the ability to predict indirect protein-protein interactions versus direct protein-protein interactions. So a good example of this is, for instance, if we look at random forest models trained on the statistical information from modes 1 to 50 of the eigenspectrum, what we find is that these RF models are particularly good at being able to predict indirect protein-protein interactions, but are particularly bad at being able to predict direct protein-protein interactions. This is completely consistent with the idea that direct protein-protein interactions live at the bottom of the spectrum, but indirect live more proximal towards the shallow end of the spectrum. And it's worth pointing out that I think because we were able to disentangle and deconvolute these admixed signals, our RF models were actually able to classify both simultaneously. So training random forest models on what we were calling mutual information window spectral correlations, MIWSCs, uniquely enabled predicting E. coli K12 protein-protein interactions at different scales of biological organization, both indirect and direct. So what about for other bacteria? the reason that we asked this question is because the spectral correlations that we computed weren't E. coli specific. It's not like we took E. coli, deconstructed it in the lab, got an observation set out, and then ran a model. We actually created these spectral correlations based on the pattern of statistical variation across the 7,000 organisms. So we thought that maybe what we trained may actually generalize, quote, out of bag, meaning out of E. coli K12 into other organisms as well. So we took the same RF models that we trained on E. coli K12's spectral correlations, and we asked whether they were good to be able to predict direct interactions across a litany of other organisms. These ranged from different phyla, across different phyla, and including an organism, Azotobacter vinlandii, that wasn't even in the initial input matrix. We compared our results to a models trained on random information, models trained on gene co-occurrence, gene neighborhood, and gene fusion, sort of the three typical ways that the string database looks for putative interactions between proteins. And what I'm showing you here is the precision of our approach for being able to predict direct protein-protein interactions for all of these 11 different organisms. And what we find is that to a statistically significant degree, our approach is able to increase the precision of other approaches, at least compared to GC, GN, and GF. So to summarize the results so far, what we found is that if we could perform spectral decomposition of a diverse data set across orthologous gene group covariation, we see a hierarchy of biological information. The shallow modes of variation are enriched for phylogenetic information, the deep modes for direct protein-protein interactions within the proteome of an organism, and the middle modes for indirect protein-protein interactions. We also find that we can use this information to be able to accurately predict protein-protein interactions within organisms. And what I'll just say in sort of a parenthetical sense is that one of the things we wanted to know was whether or not what we're finding is due to our definition of orthology. Orthologous gene group is merely one way of defining orthology across different bacteria. So we repeated our analysis, but instead of using orthologous gene groups, we used domains, PFAM domains. Domains are independently foldable and functional units of proteins. In a very similar sense, they get grouped together if amino acid sequence is similar across different bacteria. And what we find is almost exactly the same result. That is that if we perform spectral decomposition, we see a linear scaling of the eigenspectrum that's almost superimposable on that of OGG covariation. And we also find that the spectral separation obeys by the same trend as what we found with considering orthologous gene groups. Now, it's worth noting that these matrices are numerically, this matrix is numerically different from the orthologous gene group matrix. You can see that the dimensions are different. It's 7,000 by 7,200 rather than 10,000. And the numbers are actually different within the matrix, but the pattern of covariation seems to be the same.
Actually, Arjun, if you don't mind, I'd love to interrupt with a quick question there.
Yeah.
So it looks like you're enumerating, it's not just binary, like has the domain or doesn't, but it's like how many copies of the domain it has.
Yeah.
And how critical is that? Like, have you looked at all, like if you just binarize it and put ones or zeros there, how much it changes?
Yeah, it doesn't change that much. It actually doesn't change at all. So like the qualitative nature of the result stays the same. We did this very naively because we were thinking, okay, well, maybe there's more information about the relative amounts. But I think that the reason that is because I think there's just a lot of information about just co-occurrence. So meaning you can compress this down to ones and zeros, and you see some differences, and I'll get to what those differences are, but they're subtle. They're not at the scale of being able to say whether the eigenspectrum separates these scales.
Cool, thanks.
Yeah, that's a good question. Okay, so the last sort of result based on the last piece of data is that these results are robust to the specific definition of orthology. And I would say that maybe a takeaway point right now is that the whole spectrum, including components that harbor really like a minutia of data variance out to component 3000, harbor important biological information. That is non-random statistical signal that tells us about patterns of interactions just at very specified and precise scales versus those that are global scales, a la phylogeny. So if you recall the original point of all of this, though, it was to make a model of emergence. And so far I have said nothing about this. So the remainder of the talk is our attempt at trying to be able to leverage what we have found so far to create a model of emergent organization across scales of biology. So the question that we asked was, given what we've seen, could the SVD spectrum itself provide this model? And what we wanted to do was test this idea using bacterial motility as a model system. Now, the reason that we picked bacterial motility is not surprising, really. It's because it's a very well-studied pathway, but it's a pathway that is hierarchically complex. So here is a flagellum. You can see that there are lots of parts that interact together to create the collective, quote, flagellum. And we also know that the flagellum lives within a hierarchy, if we look at the KEGG database, for instance, such that the flagellum interacts with chemotaxis to be able to produce directed motility of an organism. So what we wanted to do was wonder, well, could we actually just derive this purely statistically from our data without knowing any of the biology or without knowing any of the underlying functions or structures of any of these modules shown in KEGG? So here was our approach. We know that one particular part of the SVD spectrum specifies indirect protein-protein interactions, pathway level interactions. And so what we wanted to do was define the proteins that co-evolve with a protein that we know is the flagellar filament, fleece C. So this is the only biological information that we did use. And we wanted to define all the proteins that co-evolve with this protein at the scale of the pathway, meaning within this region of the SVD spectrum. So this is what we found. We found that if we do sort of like an in silico pull down of coevolution with fleece C, we find that there are 75 proteins that co-evolve with fleece C. And you can see that a lot of these proteins are enriched in their name with respect to flagellar mediated or chemotaxis mediated proteins. So key W, key Z, key R, a lot of flige proteins, etc. So this would be the parts list that we have. And now we want to convert this into an interaction graph. So we introduced a metric called spectral depth, which is the depth in the SVD spectrum up to which two proteins are spectrally correlated. So what we mean by this is pictorially represented here. What I'm showing you are traces of the spectral correlations between pairs of proteins, and those pairs are shown here. So if we look at two examples, we get a sense of what spectral depth means. So if we look at MOT A and MOT B, these two proteins physically interact to create the motor complex of the flagellum. And what you can see is that they're spectrally correlated to a significant degree all the way out to here. This is the first point where they basically start going below a noise threshold that we computed for spectral correlation. So the spectral depth of MOT A and MOT B is deep. If we compare this to MOT A and key R is a chemotaxis related protein. Both of these proteins indirectly interact, as evidenced by the KEGG database, to produce the collective function of directed motility, but they don't physically interact. And what we can see is that the spectral depth of correlation accurately reflects that characterization, where MOT A and key R have a shallow spectral depth. So what we did was we computed the spectral depth for all pairs of proteins across all of the proteins. That should say 75, not 50. And we then thresholded according to spectral depth. So what I mean by this is we took this pairwise spectral depth matrix and we said, okay, for a threshold of a spectral depth of 50, are these pixels above or below 50? if they're above 50, that means that the spectral depth is shallower than 50, so that pixel goes to 0. And if it's deeper than 50, that means that the spectral depth goes on beyond 50, and so the pixel is a 1. So what you're seeing here is actually the thresholded matrices from 50 down to 1000. What you can see is that at a spectral depth of 50, everything is correlated. And at a spectral depth of 1000, that matrix becomes quite sparse. You can see that there are a few off-diagonal terms, but most of it's white. So what does this look like from a network graph perspective? At a spectral depth of 50, we see that everything is correlated. So we basically see a hairball. All of these nodes here are one of the 75 proteins that we had shown before. And any edge here represents whether two proteins statistically interact with each other. And if we perform gene set enrichment analysis on this, what we find is that there's a substantial ontological enrichment for flagellar assembly or directed motility. Okay, so here's the interesting thing. So if we go down to a spectral depth of 300, what we see is that the hairball of an interaction graph now fractures anisotropically into 1, 2, 3, 4 different interaction graphs. And if we perform gene set enrichment analysis on this, what we find is that these networks are substantially enriched for the flagellum, for chemotaxis signaling, for cyclic di-GMP signaling, and for LPS biosynthesis. And if we proceed even deeper, what we find is that these modules, like chemotaxis signaling, for instance, now splits up into chemotransmission, chemoreception, and signal transduction. You see that cyclic di-GMP signaling now splits up into cyclic di-GMP synthesis and catabolism. And we see that the flagellum now splits up at a spectral depth of 1000 into the parts, the individual interactions of proteins that create the flagellum. What's also worth noting is that all of these ontological terms are statistically significant. So though the significance is not changing, it's not becoming insignificant, the ontological term is changing. So if we put these together, what we see is that we were able to go from the genome of E. coli K12 to a model that represents a hierarchy of interactions that describe at the very bottom each individual protein, the bottom layer here, which is a spectral depth of 1000, the complexes, how those complexes integrate into the middle layer to create macro complexes or signaling networks, and how these interactions now integrate to create the collective function of directed motility. Now, it's worth noting, right, this is a purely statistical description. There's no biology that goes into this, but there's a substantial overlap with the biology that we know. We wanted to quantify this in some way. This is a difficult thing to quantify, but we at least qualitatively compared what we found, our statistical hierarchy, with the KEGG hierarchy. And what we find is nearly a one-to-one correspondence. What you're seeing here in the middle is the Venn diagram of overlap between proteins found on the left and proteins found on the right, our model versus experimentally derived models. And what's worth pointing out is that not only do we see the same sort of architecture, meaning that the same units of the flagellum integrate to create the flagellum, or the same units of chemotaxis integrate to create chemotaxis signaling. We also see that chemotaxis signaling and flagellum integrate to create a collective phenotype, but we also see that there are some unknowns that weren't included in KEGG. And indeed, if you look in the literature, you can find substantial evidence for cyclic di-GMP signaling and LPS biosynthesis affecting flagellar motility in E. coli K12. So it seems that our approach describes emergence across at least this pathway and this bacteria. And what I'm not going to show you just for the sake of time is that we were able to show that this is generally true. So if we look at B. subtilis, or if we look at Pseudomonas, we are able to describe the same sort of hierarchy of motility. Also, if we look at non-physically coupled pathways, like if we look at amino acid metabolism in E. coli K12, we're also able to pull out a similar sort of hierarchy that matches very well with the KEGG hierarchy. So The last thing that I'll say is it's remarkable in some ways that we were able to create a statistical representation of a hierarchical architecture. What you can see from this is that we are finding proteins that may not be in current databases or that may be connected with proteins of known function but are uncharacterized. And so the last thing that we wanted to do was ask whether or not this kind of approach, a purely statistical approach, could be used to discover new biology. So we looked at motility in Pseudomonas aeruginosa as a model. And the reason we did this was because there are a substantial number of uncharacterized proteins in Pseudomonas, at least relative to E. coli K12, which is very well characterized. And there also exists robust assays to be able to test different types of motility in Pseudomonas. So we created the same sort of network graph. Instead of using FLE-C, the flagellar filament, as a bait, what we used here was PIL-A, which is a core structural protein for Pilus, for Pilus-based motility in Pseudomonas. And this is the network graph that we found at spectral depth 50. So again, what you're seeing here, each protein is a node, and each edge means that these two proteins statistically interact with each other. At a shallow depth in the spectrum, what we see is that everything is connected. And if we perform gene set enrichment analysis, we see substantial enrichment, not for any one term, but for many different types of terms, many different terms that describe overall motility phenotypes: methyl accepting chemotaxis, cell motility and secretion, two-component system signaling, type 4 Pilus-dependent motility, etc. One other thing to note here is that there are 141 proteins in total, 70 of which are uncharacterized. So if one now wanted to do the experiment where you said, well, this graph will help me determine what the function of an uncharacterized protein is, it's still a lot of complexity to do in the lab, right? But here's what was pretty interesting. So as we spectrally filter down to spectral depth of 300, what we see are a similar pattern to what we saw in E. coli K12, that we get these distinct modules of statistically connected subnetworks. And so I'll point out three of them here. One is methyl accepting chemotaxis, the other is Pilus-based motility, and the other is flagellar-based motility. So we use this as an opportunity to say, well, what we're seeing now is the specification of a global function, motility, into specific types of functions, Pilus motility or flagellar motility. And if we look at the Pilus motility network graph, what we see is that there are uncharacterized proteins within this network graph: Q9I5G6, IOG2, IOG1, I5R2, and you see PIL-A here in blue. So we thought that this was a good opportunity to test the idea of whether or not these proteins that are, for all intents and purposes, uncharacterized. I think Q9I5G6 has one DUF, domain of unknown function. Whether or not we could assign a function to them and experimentally validate this function. So the assigned function would be they should affect Pilus motility, and what we wanted to do was test this in the lab. Importantly, they should also not affect bacterial flagellar motility. So what I'm showing you here is one of these results. So we ordered transposon mutants for all of these uncharacterized proteins. We assayed these transposon mutants for whether or not they affected Pilus-based motility or flagellar-based motility. And then we asked, if they do, then if transcomplemented by the gene, does it rescue? So transposon mutagenesis is heavily dependent on the site of where the transposon actually inserts. So we ordered multiple transposon mutants of these genes. And what I'm showing you here is just one example of the results that we found for Q9I5G6. So what you see on the x-axis here is wild-type PA01 Pseudomonas, one of the transposon mutants for Q9I5G6, the other transposon mutant for the same gene, and then the transcomplementation of the gene in the background of the transposon mutants. What you see in the bar graphs are the measurements of twitch motility as a function of 24, 48, or 72 hours post assay induction. So the wild type dynamics look like this. What you can see is that Q9I5G6 mutants abrogate the dynamics of twitch-based motility. And you can see that complementing rescues back twitch-based motility. What you're seeing in the inset is the effect of transposon mutants of Q9I5G6 on bacterial, on flagellar-based motility. So what we found is that relative to wild type, mutating these does not affect flagellar-based motility. And here are the crystal violet stains to back up what we're showing here. So the result from this was to say that this was a way that one could imagine using this approach to be able to discover new biology in a potentially scalable way. We were able to whittle down the complexity of the entire PA01 genome down to a set of on the order of 70 genes based on a shallow spectral depth. But then as we went further down in spectral depth, we purified or coarse-grained the network graph into sub-networks that were able to provide us more information about what experiments to do. So what I'll end with is saying that our approach, we call it spectral correlation analysis of layered evolutionary scales, appears to yield a valid model of emergence. That is, how proteins at the quote bottom layer integrate together to create protein complexes, and then ultimately pathways that ultimately influence phenotype and fitness. And what I would say, generalizing our approach, beyond the acronym that we created, underlying what we did was the idea of moving away from interrogating individual systems in the lab, meaning taking apart, say, E. coli, K12, or Pseudomonas, and rather leveraging patterns of genomic constraint across extant diversity to be able to infer what patterns of interactions are important for function. So what we're actively trying to look at in the lab right now is how general is this result? What we found is that the whole spectrum of evolutionary covariation describes how individual components of a system can organize to create collective structures that encode collective functions. One can imagine that this is actually a potentially, general framework to describe systems across biology. Proteins are made of amino acids, cells are made of genomes, ecosystems are made of taxa. We go out and sequence these things, and we get a parts list. And then trying to understand how this parts list encodes function is something that we believe our approach could help with. So what I'll end with is saying that perhaps scales could be a transform for describing a, quote, wiring diagram for how individual parts create collectives. And we're testing whether or not this is ubiquitous across these different scales of organization and biology, as Kim alluded to in the introduction. So with that, I'd like to thank the lab, and particularly Mark Zeidman, who is a pathologist at WashU, who really led the charge in doing a lot of the work that I presented today. Thank you very much.
Okay, thank you for a wonderful talk, Arjun. We can have a moment for a few questions if we like. While people in the audience are thinking, I've got one for you. So I think it's, kind of thinking about your results with motility, right? And looking at hierarchical organization across the flagella from like different bacterial species. I guess my understanding is because you're using kind of the same underlying correlation matrix that you'll find kind of a similar hierarchical structure, whether you're looking at like Pseudomonas versus E. coli. And have you thought at all about, or is there a way that one could do sort of a differential analysis to try to understand how like motility in one set of like species or clade might differ from motility in another clade?
Yeah, I think that's a great question. And we are in the midst of trying to do this. So I don't think it's as, I can shed light on a little bit of what I think may be an approach. I don't think it's as simple as, say, taking one and subtracting the other, because I think ultimately what we are finding is that there's kind of like a a tree-based embedding that is defining the relationships between all of these organisms. So another way of saying this is what you just said, like what is the difference between motility in one organism and another? You'll probably see some gross difference, but the path of getting from one to another has to go through some like broader intermediate and then go to the differentiated one. So yeah, I think that we've thought about doing this because the implication is that you should now be able to define, in this case, strain level variation very quantitatively. You see it actually. So if you look at B. subtilis versus E. coli, B. subtilis doesn't have an LNP ring, and E. coli does. And it comes about because the presence of an LNP ring is coupled to whether or not you're gram positive or gram negative.
Okay.
So there's an instance where just subtracting the two doesn't actually give you the full picture, because you probably have to go to some clade that's like, are you gram positive or gram negative? Then go and say, you do or do not have an LNP ring.
Mm-hmm. Very cool.
Yeah.
I guess another question that I mean, at this point, I think it would be sort of like speculation, but I mean, you and I have talked before about the idea that these cellular systems are kind of modular and therefore parts might interchange. If you were to kind of guess for, say, like these decompositions, do you think you have kind of a clear sense of at what level you might be able to sort of shuffle or swap parts between different species? Or like, do you think there's a clear break point where it's modular versus not, or do you think it's more kind of nuanced than that?
Another good question. I have a hard time answering this because we've in our lab we've talked about this very problem, and I think what our results are showing is that modularity depends on where you are phylogenetically. So in other words, like if you have E. coli K12 and E. coli K13, I don't even know if that exists, but let's just say that there's something like that exists, right? Then you can imagine that it has the ability to move, and it has a flagellum, chemotaxis, and GMP signaling LPS biosynthesis. But then at the very bottom, there may be some very nuanced difference that is the modular part. Mm-hmm. But now if you compare, E. coli K12 to something distant, like let's say Pseudomonas, okay, and you may need to go further up in this protein-protein interaction graph and swap out parts, because the thing is phylogenetically different. So you have to respect levels of phylogeny when trying to define modularity. So, in 99, I think Leibler and Murray had this paper where they were like, we need to go away from molecular biology to modular biology. Okay. Yeah. I think that there's I agree with that sentiment. I just think that there needs to be some refinement about what we exactly mean. And I think these data would suggest that it's intrinsically intertwined with relatedness.